Welcome to the South Fork Sea Farmers Present. Today's topic is a blueprint for bay restoration. And our guest is Dr. Chris Gobler. Welcome.
Thank you.
I'm going to go through some of your credits. We could probably spend most of the first 15 minutes on that. But the Endowed Chair of Coastal Ecology and Conservation, School of Marine and Atmospheric Sciences, Director, New York State Center for Clean Water Technology, Stony Brook University. And you also have three, what I found very interesting, current projects: the Long Island Shellfish Restoration Project, the Shinnecock Bay Restoration Program, and also the Georgica Pond Project. And I'm sure we'll touch on each of those as we discuss. But I'd like to start by showing people your website,
Okay.
the Gobler Lab, if you could bring that up. And if you just scroll down to the goals, which is about halfway down the first page. Okay, perfect. The first goal, understand how, see if I say this right, anthropogenic activities alter the ecological and biochemical functioning of coastal ecosystems. 2, discover means by which impaired functioning of coastal ecosystems may be mitigated. And 3, share our understanding and discoveries with scientists, policymakers, managers, and the public. So if I restate, okay, that first goal, the term anthropogenic, human activity and how it causes pollution.
Yep.
Is that correct?
Correct. Yep.
Okay. So let's start with that.
Okay.
Okay. When I moved here about 10 years ago to the East End from Up Island, as they say, I all of a sudden became very connected to the water. I spent a lot of time kayaking and just wading through both Accabonac Harbor and also Napeague. And at first, I was like, how nice this is, how clean the bottom is.
Yeah.
And then the more I thought about it, the more I was like, the bottom's a desert. There's nothing growing. It was nice to walk on, but I saw very little life. So let's start with, how did we get to this state in our bays? What caused us to have basically a desert under the water?
Yeah. Well, the deserts aren't everywhere, but certainly you're on the mark. There are locations that have turned into deserts. And there's a lot going on all at once. So we know, for example, there's been things like the overharvest of shellfish. We know that climate change is progressing and has all sorts of impacts on coastal ecosystems. And we know we just have more people in our all of our what we would call watersheds around here. And more people means well, it means many things. But as I most commonly think about it, it means more nitrogen going from land into sea. And that has a big cascading effect on all of our coastal waters.
So what does nitrogen do? What's so bad about nitrogen?
Yeah, well, I mean, on the one hand, it's a key nutrient, right? So, you know, we can't survive as individuals without nitrogen. It's what makes up protein, for example. And the same thing could be said for marine life. But of course, like everything, there's the old term dose makes the poison, and you know, everything in balance. And so in certain locations, you know, a little bit or even a lot of nitrogen won't have a big effect. But in estuaries, where the water is not circulating very quickly and the nitrogen can be retained, that can manifest in ways that are detrimental. For example, degradation of salt marshes along the shoreline. And once it's out in the water, it can stimulate things known as algal blooms. Those algal blooms can be harmful to humans or marine life and
So
the
these are
habitat.
the red tides, brown tides, that's what we're talking about?
Exactly. And you know, and those have cascading effects. You know, if they get too intense, they can shade out seagrass beds, and that's a critical marine habitat. If they get too intense, they can lead to the die-off of shellfish or fish. So, you know, and it all stems, you know, stepping all the way back, you know, more people in the watershed, and, you know, I didn't get into the detail when I first introduced it, but, you know, here in East Hampton, and actually, frankly, for much of Suffolk County, everybody has septic systems that are purposely designed to take wastewater from the home and inject it right into the groundwater without any treatment. And so when you do that and you have more and more people, that's more and more septic systems and more nitrogen that, in short order leaches out into surface waters.
So the septic systems, if we were still the sparse population of the 40s out here,
Mm-hmm.
probably would be okay or not as detrimental.
Yeah, like I said, dose makes the poison.
Right,
So
right.
just a few people and a few septic systems, sure, that probably wouldn't be of any concern. But you add in more and more of those, and over time, that's what's led to the sharp rise in nitrogen and also, and the cascading effects thereafter.
Now, on the bright side, your second goal, which was to discover means by which impaired functioning of coastal ecosystems may be mitigated,
Yeah,
you spent a lot of time working on that.
correct. Yeah.
And basically, that second goal is, what can we do about this? How can we not turn back the clock and get back to what was, but create a healthy ecosystem?
Yep, yeah, and there's many ways to go about that. I think what we'll talk most about is what we call the in the water solutions, or I would call. I'll just mention before we dive into that as director of the New York State Center for Clean Water Technology, what we're focused on is the creation of new septic systems that remove the nitrogen before it goes into the groundwater, and also remove other things that we don't want in our drinking water. Right?
Mhm.
We forget sometimes it's not just the effect on the bays, but gee, that groundwater is what we drink every single day. Even if you have what people call city water, or really it's water from Suffolk County Water Authority, it's coming right out of the ground and not far from where you live. And so we want to get it's not just the nitrogen to get out in that front, but it's also drugs, pharmaceuticals, personal care products. And anyway, at the Center for Clean Water Technology, we're working on systems that not just remove the nitrogen, but all those other compounds as well.
And correct me if I'm wrong, but Long Island is fairly unique in that it has this aquifer, which is basically a bubble underneath us,
Yep,
and that's our only fresh water.
yep. Sole source aquifer.
Right, it's not coming
And,
from anywhere upstate, it's right underneath us, and that's it.
Yeah, precisely. And the closer you get to the shorelines, so further you get out on the forks or closer to the watershed here, that aquifer gets smaller and more prone to saltwater intrusion, shallower and such. So and therefore collectively more sensitive.
Right.
Yeah.
Okay. So let's get into your work then with mitigation.
Yeah.
I was really excited. We've had other guests on talking about your work in Shinnecock Bay.
Okay. Yeah.
And that just seems so exciting to have a place where we can say, look, it can work.
Mm.
So try to, walk us through that. What have you done that's been successful there?
Sure. Well, that project is literally a decadal project. So, my first faculty position was Southampton College, right? And that's the Southampton campus. I'm actually still there, but now Stony Brook runs the show.
Mm-hmm.
But started there in 1999. So the first decade or so was understanding and identifying the problem, which everything I just described to you before is what was happening there. But concurrently, also thinking about what might be some solutions, and specifically looking at what's the current state, for example, of filter feeding bivalves. These are clams, oysters, mussels, scallops, organisms that filter the water, and like a pool filter or a filter on a fish tank can keep that water clean if they're at high enough densities. And what we found way back then was that the densities were so low that there was no way they would have any effect in filtering the water. And even worse than that, they were so spread out that their prospects for coming back were almost nil. Bivalves are what we call broadcast spawners, as are many marine invertebrates. So to reproduce, they throw their gametes in the water, and success is when those gametes collide. But if you have a clam way over there, and then the male's over there, the female's over there, they spawn, and the gametes will take
Right.
the tides will take those gametes away, and so it was what we called at that point, scientifically, it's called a recruitment-limited population. So recruitment is the idea of bringing new recruits, new young into the population. Inability to have high rates of successful fertilization, reproduction would make recruitment not really a prospect. So then, knowing that, we considered, okay, well, maybe what we need to do then is to build back that reproductive population. So we did a lot of research trying to understand where in the estuary would be a good place to do that. And then also designing what we called spawner sanctuaries. So we firstly, I should mention, we landed on, go ahead.
When you said finding the, excuse me, the right place,
Yeah.
you had to study the currents and the depths and see where, when they did spawn, where it was going to go? How do you go about that?
All those things. So firstly, we needed to make sure the bottom of the bays were suitable for the, if we're going to put new adults in there, is it the right kind of sediment? Secondly, for the water quality was there. Would the water quality be of such a level that the adults could reproduce? In some places the water quality is so bad that the adults literally can't get up the physiological capacity to reproduce. So good bottom type, good water quality.
Now, back to good bottom just for a sec.
Mm-hmm.
Good bottom, we're looking at, I think, mostly clams and oysters when we talk about these projects.
Yep.
Good bottom is different for clams and oysters?
Yes, very different actually, because oysters are colonial individuals that like to attach to a hard bottom, or actually have to attach to a hard bottom. Whereas clams are, pretty flexible on that front. There's if it's the organic muds that are too organically enriched, or those smelly sulfitic muds, that's probably not good. But and actually, if a very hard bottom is not good for a clam either, they're what we call they dig into the sediment, and so they need to have it a little soft to be able to get in there. And also, we didn't want a ton of seaweeds, which can overgrow. There are certain places where the seaweeds are so thick they can actually overgrow the bivalves, and that's a problem also.
Okay.
But yeah, good bottom type, good water quality. And then you mentioned, currents. That was critically important also. We knew that if we put the bivalve, say, right by in Shinnecock Bay, it's the Atlantic Ocean, the Shinnecock Inlet, which is the inlet to the ocean, they'd spawn, and then on the tide, their young would go out to the ocean, and, that would be the end of that. And then conversely, if we put them too far to the west, dominant currents might transport them into the next system, which would be Moriches Bay. So, it really was a matter of finding a sweet spot where, they would be retained in the bay, and again, the conditions would be permissive, not just of the adult reconditioning, but the other aspect is permissive of their young surviving. We also knew that there were some places where the water quality was so bad that, yes, they might recondition and spawn, but then, the larval stage, which is the first stage once they reproduce, would not survive. And then once we found those locations that were permissive, the other important thing was actually packing in lots of clams very close together, right? So that when they would spawn, it would be successful. So when we first looked at the densities of clams, it was less than 1 clam per meter squared. So we were shooting to get about 20 clams per meter squared, and logistically that ended up being about 50,000 clams in about a half-acre plot.
So 50 per meter squared just in the sanctuary area?
Correct.
Okay.
20 per meter
20,
squared.
yes.
Yeah, 50,000 clams in a
50,000.
half acre. Yeah.
Okay. Now, how was it getting a sanctuary set? Getting
Yeah.
the local powers that be to agree to, nobody's going to take the shellfish from this area.
Yeah. Well, there's the old saying, it takes a village, and so in this case, it took a village, a town, and a state. So we had to work with the New York State DEC to have them permit what we were doing. But the town of Southampton, like East Hampton, there's the trustees there, and so I went to the trustees and said, what, I'd like to. These are the areas we've identified would be ideal for planting clams. we want to do that and make sure that no one can harvest the clams. And they said, great, on one condition. You have to come with us and go to the Southampton Town Baymen's Association meeting, and you have to tell them what you're going to do and why. And so, that was a little bit of a daunting meeting. So we went there, and I explained what the plan was. So, the first step of that explanation is, so I want to shut down these, whatever, hundreds of acres to shellfishing. So, Dower looks in the room. But as I began to explain it, actually there was buy-in, and they said, okay, if, and there was some negotiation, like, I had a line here, they said, well, let's move that line up a little bit, because actually we do like to harvest there.
Right.
But in the end, frankly, any success is owed, it's not owed to me, it takes a village. And so it was because the Baymen got on board, and the town got on board, and the state got on board, and frankly, the Baymen ended up being some of our best advocates and best allies, because they're the ones on the water. So we had multiple times where we'd have Baymen who would approach people on the water and say, hey, this is a sanctuary, you can't be harvesting here. And I know that because actually sometimes they'd approach my students. They'd be out there monitoring. They'd have a rake, and it looked like they were commercial harvesters, but really they were obviously just sampling to see how the clams were doing, checking survival and such. But yeah, it was the partnership that made it work.
Well, the Baymen are the one who understand the life cycle of the clam, and they're if we get a healthy bayback, they're the ones who are going to benefit.
Yeah.
Well, we're all going to benefit, but their livelihood will benefit.
Yeah, and in the beginning, so I said we started 20 years ago, so it was 10 years ago we started the project, is when I had to have that conversation. And, everything I was saying there were it was speculative, promises, like, well, if we do A, we think we'll get B.
People love working on ifs.
This
might work out, but
fallback plan and contingencies and such. But in happy news, that project, the Shinnecock Bay Restoration Project, was funded by the Lori Landau and Bob Mays Foundation largely, and they also were great partners and supportive, and importantly kept up the support for a decade. And I emphasize that because it took that long, almost, to see all the results. The results trickled in year by year, and in the very beginning it looked like nothing was happening, and then all of a sudden you start seeing some small clams, and you say, oh, you of course, on the one part of me wants to say, oh, that's it, we did it. But then the scientists and me and my colleagues would say, well, let's wait and see, and we've got to get more evidence. So it took time, and same with changes in water quality. We started seeing water quality improving, but you look back over the years, there can always be a random good year where, oh yeah, it's better this year, and then it falls back the following year. So it did take, frankly, it took 10 years of convincing ourselves that this was really happening. And then, the 10th year was this year in 2022, and we just published a paper in the Frontiers of Marine Sciences, free online scientific publication, international peer-reviewed publication that anyone can access that documents everything that we found.
And you have some slides, I think, that are good graphics for the results you have.
Yeah. We can go through some of those. If you want to
let's take a look at some of the slides now.
Okay. I think we're going to have to zip ahead, though.
Yeah, we've kind of covered that.
Yeah.
Next slide.
We can skip through probably the first few.
Next slide.
Yeah.
Can we talk about this slide for a second? Because
sure.
I think people need to understand. Can you? Because I know this might be impossible. Explain this slide in a couple of sentences, because it's got a ton of information.
Yeah. So this is a slide or a map that my lab puts together every single year, and it looks like a lot of data, but it's actually all collected in 4 months. So this is only the months of June through September, and it's the documentation of harmful algal blooms and hypoxia and fish kills on Long Island from June to September 2022. So we're monitoring, dozens of locations every week, and our lab we do our own active monitoring, and then in addition, we get samples brought into our lab. And we make measurements. When we're making measurements, we're comparing them always to state and federal guidelines. So there are state guidelines for oxygen. There are state guidelines, for example, for blue-green algae. And then there's even, scientific levels of for harmful algal blooms that you would document. And, a fish kill is a fish kill. But so that's what essentially this shows, and really documents makes the case or demonstrates that there's a lot of water quality impairment to be had. And the one really interesting thing is if you look at there's like two areas maybe where there's no water quality impairment. One is, you could say, the northeast part of Long Island, eastern Long Island Sound, and there's lots of tidal flushing there, and not a lot of homes relative to the very large water body,
Right.
Long Island Sound being the third largest by volume in the nation. And then the southwest corner, that whole area is sewered, right? There's no sewage going into the water from on-site septic systems. Where everywhere else, the entire North Shore, including Nassau County, most of the nitrogen's from septic systems, and the rest of Suffolk County as well. But anyway, that's sort of
And
an add-on.
this map is used by a lot of municipalities to report to their constituents the state of their base.
I think, in some cases.
Yeah.
Yeah.
I've seen it other places. so,
Okay. Yeah.
Cool. Next slide, please.
And this is just the fact that in addition to the fact we have the local issue going on, there is the global issue of climate change as well. And people most commonly may think of rising temperatures, and that certainly is a very big issue.
Mm-hmm.
But hand in hand with that is the concept of ocean acidification, that the cause of climate change is carbon dioxide, right? And that's what then leads to the greenhouse effect and rising temperatures. But it actually has a direct effect on water chemistry, driving down the pH, and that has consequences for marine life.
Making it more acidic,
Correct.
and that is probably bad for shells.
Exactly. It just makes it makes it more difficult. In fact, I think on the next slide, perhaps, even if we advance to
Next
that
slide.
one.
Next slide, please. Thanks.
Yeah, so this is just experiments that we had published, just essentially showing as you increase the levels of CO2, these juvenile hard clams and scallops just get smaller and smaller and smaller. And this is just these are what we call the survivors. There's the same effect on the survival rates are lower at the higher levels of CO2 as well.
Are they most susceptible at the larval stage when they're trying to grow their first shell? I would
Exactly.
imagine that's
No, you said it exactly right. In
Okay.
fact, that's you can think of it that when they're first spawned, those larvae are naked. They have
Right.
no calcium carbonate, and therefore it's a huge effort and to get the constituents out of seawater. Once they have a basal shell, that can serve as a buffer against the acidification. So if they're in bad conditions, they can actually rely on their own shell they've already made to buffer against the acidification. But as larvae without any shell, that's like this critical stage. So and that these experiments were on larvae. So this is like 40 days post spawn.
next slide, please.
Okay, so now this is getting into the Shinnecock Bay project.
I knew we'd get back there.
Yeah. So no title on this one, but this is actually this is the circulation of the water. So A and B are the two locations where we put the spawner sanctuary. So this yellow stars there. And then superimposed on that is where would the larvae move, or where do they move post-spawn? So in A and B, what you see is they stay largely in the bay, a little bit of trickling out of the inlet. But by and large, they're staying in the bay and populating both the eastern and western part of the bay, depending on which site.
Mm-hmm.
And then C is just to show that anything spawning in the eastern part of the bay, they all go out the inlet. So you'll note there's a lot fewer dots, and that's because after just 48 hours, all the larvae are evicted out, and that's irreversible. They don't come back. Because part of what we actually ended up doing is increasing the populations to the east. So people would say, oh, well, that population is just recreating itself, reproducing, and replacing itself. And actually, by laws of physics, that's not possible.
So that whole large area adjacent to the inlet basically washes out with the tide.
This, yeah, exactly.
But by going off to the west a little bit, you minimize that effect.
Exactly.
Cool. Very nice.
And this is a little complex, but on the top it's showing the spawning of the clams, and on the bottom it's showing the actual larvae. So you can see the top one's called gonad rank. So essentially the larger the number, the more ready they are to spawn, and when the number drops, it indicates that they actually have spawned. So you can see in that July period, the number drops from around 3.5 to around 3, and then it comes back thereafter. But on the bottom, it's showing the population densities of the larvae in the water.
Okay.
And essentially, if you look at the two together, you see that there's the drop in the gonad rank, that means they've spawned, and then there's the larvae in the water column. And if you really parse out the locations, the first peak is where we planted the clams, and then the ones thereafter are by the bridge, so the eastern part of the bridge, the western part of the bridge, then the eastern part of the bridge, and then actually within eastern Shinnecock Bay. So exactly as the models suggested, the larvae are spawned in the spawner sanctuary and then move over to the east.
And how long is the larval stage until they actually set and become what we would identify as a clam and
Yeah.
stop burrowing?
About 2 weeks.
So that's a long time to be
Exactly.
basically subject to the tides.
Right. Yeah.
Okay,
Yeah.
thanks. Next slide.
Yeah, and so this is the tidal area cutoff. A bit says change in densities of hard clams by size in Shinnecock Bay.
Mm-hmm.
And so what you see is, probably the first thing that jumps out is the eastern part of the bay is the most dramatic. Right? And so what we see there, if we start there, and again, that's where we know that the larvae are being transported to, you see not only the total numbers go up, but if you follow the colors, you know, the largest clams aren't really changing much. But what we're seeing are all these small clams. So those are the new recruits that are coming in over time and settling. You see the same thing in the west, but it's a lot more muted. And we think that's because, you know, two things. One, there's preferential transport to the east. And then two, the water quality is still improving in the west. So it's not nearly as good in the west compared to the east. But we do think over time that we can... we'll see more of that pattern in the west as
So
well.
the Eastern Bay, because it flushes out every day, has better water quality,
Yeah.
but more difficult to use as a sanctuary because of the same reason.
Exactly. So and so, you know, sometimes I refer to those spawner sanctuaries as the Goldilocks zone, right? So you don't want to put them in the terrible water quality, right?
Right.
But you don't want to put them in the perfect water quality either. One, in this case, because they'll flush out. But just speaking more generally, you know, those are the areas that don't necessarily need the more filtration pressure. So.
Right. Next slide.
And now this is the landings. So this and a completely unexpected outcome of the project. So, you know, we the goal was to boost the densities of clams in the bay. That would increase filtration in the bay. It was not first and foremost in our mind that this would have an economic effect, but what you can see is that the increases in the harvest, and again, it's in both sides, but way muted in the west, are, you know, through the roof. If you go from when we started in 2012, I think the increase is more than 20-fold in the harvest of clams in Shinnecock Bay. And I should say, and I'll show momentarily, but you know, the harvests now exceed all other South Shore bays. And we've done some actually economics on this. If we assume that you know, we first started having harvestable clams in 2015, and we say everything before that was, you know, not due to our project, but you know, due to just maybe a random change. And in fact, you can see the slope changes markedly in 2015. You know, that harvest between 2015 and 2021, more than $3 million in on-site economic benefit just to the baymen. And then there are these economic multipliers you can put on top of that would suggest that it's maybe $30 million you put in wholesalers and markets and restaurants. So this is something we hadn't had as a focus, but clearly was a happy outcome of what we were doing as well.
And makes it much easier to take this idea and transport it to other places because you have some data that says it benefits everyone.
Yeah.
Fantastic. Now, you've mostly focused on some more change in.
Yeah, so this
hard
is just
claims.
this is the long-term trend in the landing. So what we just were looking at there was the back end of the tail.
Right.
So essentially what it shows, if you follow the 2021 number and you go all the way over, it's like we got into a time machine and we're suddenly back in the 70s, which
Right,
is,
late 70s
it
is the
was in the
last-
70s when Long Island was known for producing 2 out of every 3 hard clams eaten in the United States.
Wow.
And so, these are very substantial changes, therefore. And I think maybe the next one might be linked to this as well. if we
Next
go-
slide, please.
And so this is just showing the different South Shore bays on Long Island, just to show that, we're doing the restoration in Shinnecock Bay, and that's where the changes are. And the other places are fairly static. And you can see now, when we started, Shinnecock Bay was the worst, had the lowest yields of clams, and now exceeds all other bays. And that's despite the fact that Shinnecock Bay is 1/10 the size of Great South Bay. And there's still people actively harvesting, in all these
Oh, yeah.
systems.
Yeah. Yes. Now, you also have worked with oysters.
Correct.
And so talk a little bit about that because I'm always wondering, clams, oysters,
Yeah.
which one,
Yeah,
or should we be doing both?
Yeah. I think there's just a couple more. I
Oh,
don't
I'm
know if
sorry.
you wanted,
So,
and then we can talk about
Yeah,
the
yeah,
oysters for sure. Happy
okay.
to talk about those.
Go-
But,
I'm sorry, back to the slides. Okay.
And so this is just, I think there's just 2 on the water quality effects. So this is levels of brown tide in Shinnecock Bay over time. So, this data set goes, I was actually looking at this data set. The bloom started in 1985. They happened every single year. And this is just showing over time they've whittled away. We had no bloom again in 2022. So, and what we also were able to show in the study is that with more clams, the water's being filtered quicker.
Quick, people always worry about this. I always talk about clams and oysters as filter feeders.
Yep.
They take the bad stuff out of the water.
Yep.
And people always say, I don't want to eat that.
Yep.
How come they're still good to eat if they're
Yeah.
taking the nitrates out of the water?
Right. Well, again, nitrate is nitrogen. Nitrogen makes protein. Protein is what, is a nutrient that you get out of the clams. And so, the DEC regulates shellfishing in New York State. So they're testing all the waters for the health of the water and therefore the health of the shellfish. And they make a definite statement about every water body. It's either open or closed to shellfishing. Some of them are open just seasonally, but that's our guidance. And so, for example, there's a little bit of brown tide that gets into a clam at low levels, perfectly harmless. In fact, I should say entirely harmless because that's one of the. There's only a handful of algae that actually have a human health impact, right? Brown tide is not one of them. And in fact, most algae are actually what make the bivalves more nutritious, right? The algae actually supply, for example, the omega-3 fatty acids that everybody likes to get out of seafood. They're not being made by the shellfish; they're getting them by filter feeding the algae.
So it's really about the bivalves creating the balance and making sure there's not too many nutrients, too much nitrogen.
Correct. Yeah.
That will harm the whole system.
Yeah.
Not that what they're eating and consuming is in any way harmful to humans.
Correct. Yeah.
Anyway,
Yeah.
I get that question all the time.
Sure. Yeah. Well, you have the answers.
I think you have another slide?
Just one real
One more
quick.
slide, okay.
And that's just showing, this is a colorized map of western Shinnecock Bay. Anywhere that's green is a gain of seagrass in the bay during the period of enhanced filtration in the western part of the bay. And we only focused on the west because that's where we knew most of the seagrass had been lost and that any changes in water circulation would be due to the bivalves. And so essentially what this amounts to is hundreds of thousands of square meters of new seagrass meadows that have emerged. And we also were able to show in the study water clarity has increased. So more filtering the water, the water gets clearer, there's more light for the seagrass, and there's been an expansion of the seagrass population.
And with the seagrass, the possibility of helping the scallop population, I would assume it has to be connected.
All sorts of actual fisheries, actually. So seagrasses, people call seagrass meadows, people will call nursery habitats,
Okay.
right? So yes, scallops, but also fisheries as well, like finfish. So the small fish, they're, it's like going into the woods to hide
It's a
from
place to
predators.
hide, right.
Exactly.
Right.
And maybe even other bivalves as well. Yeah.
Cool. And that, I think we just right into the oysters.
Sure.
And that the possibility of an oyster reef.
Yep. Yep.
Also creating habitat also. So you have put oysters into Shinnecock also?
Correct. So when we began, I gave you the backstory and the story goes back 2 decades. And so when we were looking at clams, we were also looking at oysters. And, part of the effort there was thinking which bivalve would do best in what part of the bay. And we actually did, we looked at the literature, but we actually did experiments also. And, oysters are tolerant of higher temperatures. Oysters are actually more tolerant of brown tide and poor water quality. So we knew then we purposely put the hard clams in an area that was what I'd call, mediocre water quality.
Okay.
And the oysters in the area where it was actually a little bit worse because we knew they could handle it. And so, we built what the DEC tells us are the first permitted oyster reefs in New York State. So we took a long time, but we eventually got permits. And we've built, I think we're now up to like 8 oyster reefs officially out there. Although now what we're trying to do, each individual oyster reef by New York State DEC permits is 5 by 10 meters. But we're now actually starting to, put them sort of in sequence. So my colleague, who I should mention, Mike Dole, he's the associate director for shellfish restoration and aquaculture in my lab. And so he's really been an incredible partner through all of this. He was an oyster farmer actually in Montauk for 10 years. He was my colleague in graduate school in the 90s, then he was an oyster farmer, but then he came back actually to work in my lab. So he's been a great partner in all this. And so he talks about each oyster reef as a metapopulation, so that when they reproduce, they may be exchanging gametes and creating new areas, and there are new areas for oysters to recruit to. And so just a little more nuance on that. Each oyster reef is kind of like, if you can think about Legos, we make bags of oyster shell that we then spawn oyster larvae onto. So each bag or each individual shell that's in the bag can have, say, 100 oysters, right? And then there's hundreds of shells in the bag, and so that we put that out as the oyster reef. In some places, like if you were to go to Chesapeake Bay or go down to the Gulf of Mexico, oysters are so plentiful they can actually just put the shell out, and then new oysters recruit onto it.
Right.
Presently, and particularly when we started, there wasn't enough or any recruitment of oysters there, so we actually had to put the first oysters out there. And so, we set the oysters on the shell, and then the shell bags go out, and we've been monitoring them over time, and they're doing quite well. In fact, some of the oysters that we see in just 2 to 3 years are the size of your shoe.
Ooh, that's nice.
Literally the size, like, we're so used to, or at least I'm so used to, when you have an oyster in a restaurant or you buy them at a seafood market, everybody wants the little, nice little cupped oyster.
Yep.
But so we forget or don't realize that in their natural setting, I mentioned before, they grow colonially, so literally on top of each other and aggregating on each other, and in that setting grow very, very large.
And they're big and ugly.
Yeah. Which is, from a poaching perspective, is a good thing.
Yes, yes.
Nobody wants to eat an oyster the size of your shoe. Not many people.
The South Fork Sea Farmers, we just finished putting our first oyster reef in Accabonac Harbor.
Nice, yeah.
We got approval for that, and
Yeah.
we're awaiting approval for 2 more to go in Accabonac.
Wonderful. Yeah.
And one of the reasons I'm asking, because that whole concept of recruitment limitation.
Yes.
Like, is there a tipping point in volume where you start to get natural set of clams and oysters? And how would you come about? How much is needed?
Yeah. Well, firstly, I don't have a precise answer, but I think just some general guidance or things that tip things in the right direction is not just having your oyster reef, but having other populations, right, that are also in the ecosystem and reproducing, and maybe then can settle on your oyster reef, right? So it could, because it may be that the oysters on your reef, they reproduce, but those larvae may leave, right? But if there's a natural population, and probably as importantly, if there's some aquaculture going on, right? Each oyster farm is, and this is, I'm quoting Mike Dole here, each oyster farm is a spawner sanctuary. Right? It's a sanctuary because they're there and they're protected, and every year they're going to spawn, so long as they're of spawning age, and they typically are just before they're harvested. So and therefore, successful oyster restoration may well be creating reefs, having some aquacultured oysters, and then collectively those may be contributing to the natural population that starts to rebuild on its own as well.
Yeah, so what we've got going in Accabonac is it's like a triangle, that three-pronged harbor.
Yeah. Yep.
And what we want to do is locate really in almost towards the furthest extreme of each of the points.
Yep.
A reef in each point.
Yeah.
And we also have the town of East Hampton oyster gardening. So we've got probably about 30 oyster cages floating.
Great.
So we're hoping that that combination will reach that tipping point.
Yeah. Yeah.
And then it also gets seeded every year.
Yeah, well, that's. I mean, I think that is a way to go. And again, there's a lot of other regional activities also. So, you never know when a larvae that's spawned, say somewhere out in the Peconics, gets ejected in on the tide and then
Right.
lands on your oyster reef, and yeah, but that's great.
But prior to this, there really wasn't much for them to even land on. Even if we had them,
Correct.
the
yeah.
bottom was pretty barren.
That's right. That's right. Each oyster larvae is chemotaxic, so it seeks out calcium carbonate or other shells. So that's why the spat on shell works. And from an evolutionary standpoint, the idea is, well, if the adult made it there, then if I settle here, that means maybe I'll make it as well. Yeah.
Cool. The oyster reefs one more question about oyster reefs. The ones in Shinnecock, were you involved in the one that is a rectangle?
Yes, so all of that's the rectangle is our basic go-to shape, right? So the
Okay.
5 by 10.
So
yeah.
explain why the rectangle?
Right. Well, one is what the DEC allows us to do.
Getting around the
But then essentially what we generally do is the full outside is lined with the shell bags,
Mhm.
right? The inside we usually fill in with just loose shell. And then over time we may put more bags. We may put, in some cases we've put actually adult live oysters. We had a program once that was collaborative with the Nature Conservancy and Pew Charitable Trust. It was called SOAR, and I don't want to get into too much detail, but it was during the pandemic. Oyster farmers couldn't sell their oysters. They had all these leftover oysters, and so we were involved in that program that took the leftover oysters that big and craggly nobody could sell,
Yeah.
and put them and actually put those on our oyster reefs along with the the spout on shell that we already had there. But yeah, the general rectangular shape is because the outside is the spout on shell and the inside gets filled
So
in.
when getting the 50 square meters for the state, is that just the outside? And
Correct.
Then, so that, okay.
Yeah.
I'm just planning shapes of
Yeah,
reefs in
yeah,
the future.
yeah. But again, what we've done is we've then co-located those. So we started with 2, and then we started filling them in between, and went through that SOAR project, making different sorts of geometries because those weren't officially reefs. And with the goal eventually is maybe we connect all these and we have one really large reef, which is actually what we're moving towards. And there seems to be even some movement, naturally in that direction as they sort of expand and such. So yeah.
Very cool. And now, kelp.
Okay. Yeah.
Another ally in the attempt to restore our bays.
Yeah.
Talk a little bit about kelp and what it can do and how it fits in the big picture.
Sure. Maybe the best place to start with that is actually to go back to the pandemic and talk about these oyster farmers who, all of a sudden found themselves, so it's the pandemic, just making sure I'm clear with what I was saying there, restaurants are closed, they couldn't move their product, right?
Mm-hmm.
And that was actually a vulnerability that we had, was actually a motivation when we started thinking about kelp a few years prior. And that motivation is something that goes back to farming practices, right? So back in the early 20th century, Long Island land farming was a single crop. It was potatoes, right? And so if the potatoes fail, you have potato blight or something like that, then all of a sudden, what can you do, right? And so, we started saying this, we used that parallel when we started thinking about kelp. Why would you grow kelp? And there's many reasons. But the first thing we said is, well, aquaculture on Long Island needs to be diversified. If you're just relying on one crop, in 2020 it was a one-off, but what if suddenly there was a disease for oysters? And the other thing with the oysters is there's also a certain seasonality. So there's not much to do with the oysters and maintaining the oysters during winter. So when we thought about kelp, it's actually a cold water seaweed. So it only grows in the cold part of the years, and so when we're growing it for aquaculture purposes, we actually we spawn it in the fall. It goes out in the late fall, and then it actually grows from the fall and winter, and then but most robustly in the spring. And then by May we have to pull it out. It doesn't like the warm temperatures, and were you to leave it in there, it would die off. But we get tremendous yields. We've had up to kelp blade lengths in that very short time. Frankly, they don't grow much in the fall and winter. So maybe you hit like February and you're out there, and your blade might only be a few inches. You're thinking, well, this isn't working. But in the spring, as it warms up, they grow at an enormous rate. In some locations, we've got more than 10-foot long kelp blades. We put them out on rope, and in a foot of rope, we can get 10 pounds. And, we're putting out sometimes 200-foot length lines of kelp.
That puts in a foot of rope, 10 pounds of kelp.
Yeah.
Gives you,
And then think 200 feet per line. And then in some cases, we're putting out, half a dozen lines. So you can easily get to 1,000 pounds in just a few months. And everything we've done to date has really been, well, not even really, it's just been experimental scale, 6 lines. But, we in places, in the places where seaweed aquaculture is a true industry, you can have acres of kelp. And then you're looking at, tens, hundreds of thousands of pounds produced per farm in a given season.
So 2 things. What does the farmer do with the kelp? What does it
great
use?
question.
Second, what does it do for our environment?
Yeah, great. Two great questions. I'm glad you asked both, actually. Yeah. So the first question is really that's the burning issue right now. Is that I think, and foreshadowing the second one, kelp is good for the environment, and I'll detail that. So, with that as a given, how do we make that something that's also economically viable? It's not a commercial industry now. And so, frankly, there's no incentive for anyone to grow it, except for sort of a feel-good thing, and it's good for the environment. So there needs to be a lot more progress on that front. So making it a viable commercial industry. And there's a lot of effort being done by many, many groups to do that. So to think about, how do we make this a food product? So for example, right now, New York Sea Grant is working on that question. DEC is figuring out how you would regulate it as a food product. Right now, you technically you can't even sell it, but that hopefully will be fixed momentarily. But then you have to have the market, right? And you have to, and so you have to get chefs interested in using it and people interested in using it. And then along with that, there's what else can you use it for? So people have done research showing that the material within the kelp can be converted into plastics. There may be certain chemical elements you could put out of there, things like carrageenan, which could be a food additive. And then we've done actually a lot of work since the very beginning on kelp and other seaweeds and making it into a fertilizer or a soil additive. And
that seems like that's been done throughout history.
Yeah, so that you can. That's right. You can hear about settlers doing that.
Yes.
Yeah, and in some ways, I feel like we're reinventing the wheel a little bit on it. But just to add some color, we've taken seaweeds and amended them in such a way where we can make a solution. And if we test it relative to Miracle-Gro, and I just give you the numbers, you can't tell the difference. And similarly, we've done the same thing and then grown different vegetables and we get the yields, that are more than if you don't use it. And in some cases, again, if I showed you the two numbers, you couldn't tell if it was Miracle Grow or if it was the seaweed fertilizer. So, so that's another angle for it. But at the same time, that's an industry that needs to be built up. All these things do.
But now the positive effect on the environment. Why do we want kelp in our bays?
Sure, and it boils down to, most of it is because of the awesome power of photosynthesis, right? So I've actually in some of the intro slides I already showed you what are some of the problems we have in the oceans today. Well, too much carbon dioxide, right? Hypoxia, dead zones, not enough oxygen, too much nitrogen. So through the photosynthetic process, the seaweeds are taking up the CO2, giving off oxygen, and to grow, they're taking up the nitrogen like a sponge, right? So all the things that we don't want in the water, they're taken care of. And if you harvest that kelp, like I said, in May, when they're done growing, you're then what we call extracting the nitrogen. Some people would call it sequestering the carbon, and then it's taken out of the water. If it's just left there to die, then it gets recycled right back.
Right,
but if the active mode of pulling it out is remediating the bay. And so, and we've done research really showing some of the positive effects. In fact, I think if you cue to some of the slides.
Yeah, I think that we have a few slides that show some of the kelp, which would be interesting. If you guys would bring the slides back up.
Yeah, so this is just showing essentially what I just said on the chemistry front. And the only thing I didn't address is that, kelp makes bioactive compounds. It's an amazing thing. If you put...
What do you mean? What's bioactive compound?
Yeah, so all plants make what we sometimes call secondary metabolites, right? Plants, if there's a predator, a plant can't run away, right? A kelp can't run away from something that might want to eat it. And so just evolutionarily, therefore, plants and seaweeds develop certain compounds that may deter things from eating it or from colonizing on it. And it's an amazing thing when you put out kelp. You can have a line where you have these kelp blades, and they're beautiful, luxurious, maybe looking kind of like this. And then areas where there's, on the rope, where there's no kelp, and it's fouled with all sorts of organisms. And then you get to the kelp, perfectly clean, and then fouled again. Just suggesting anecdotally that they're producing something that's warding off. And I'll talk to that in a slide or so.
So people might want to wrap their boats in kelp for winter?
They'd have to, here's the thing, the kelp would have to be growing. Right? Okay. As it grows, it's producing these compounds. Yeah.
Cool. Back to the slides. Okay.
Yeah. So this is just showing. Mike Dole put together this slide, and so he's being an oyster farmer. We've grown kelp on 10 different oyster farms across New York, and also grown it out. You can see as far afield as Fisher's Island or the Bronx River Estuary, and it's grown.
I see Shell Works there.
Yeah, and it's grown great in all these locations. Yeah.
Next, please.
Yeah, and then now to get to some of the environmental benefits. There's Mike Dole on the left, and so
that kelp looks incredible.
And you can see there's nothing growing. Actually, here's the amazing thing about that kelp. Looks incredible. Note, Mike is standing, right? This is the Great Gun Oyster Farm in Rich's Bay. He's standing in about a foot or two of water. This is 10 feet of kelp blades, right? When Mike began this project, he was told, you'll never be able to grow kelp in shallow water. I won't say what the name's for, but that's not going to work. It only grows in deep water. But actually, our best yields year after year have been in this Moriches Bay site and other shallow sites. And again, because of its own natural anti-fouling properties, as long as it's growing actively, you can get, like I said, we're on the low tide. It may only be 2 feet of water, but we're getting 10 feet worth of kelp blade.
Now, how big are these kelp blades when we start with them?
Yeah, microscopic. So we set them in the lab, and so they start out, just like the larvae in shellfish. They start out just as little sporophytes that then set, and so we let them grow out on the line in the lab till you can just start to see them. Looks like little brown hairs growing on them, but it's very, very small. And then this is what it looks like in the spring, and but getting to the environmental benefits. The image on the right is the Great Gun Oyster Farm, where we did an experiment where we had kelp lines. They're probably the yellow indicates where they are. And if you look carefully, you can see some other like little U-shaped lines. Those are actually oyster bags that are part of the oyster farm. But for this experiment, we grew juvenile oysters right in between the kelp lines, right next to the kelp lines, or further away. And it was specifically, in this case, trying to understand, might the oysters do better in the presence of the kelp? So if we go to the next slide. So here we're measuring the pH, both in the kelp, or specifically on the kelp, and then the site further away. And what it shows is that the kelp has this buffering effect of actually raising up the pH. Now the pH of the water naturally should be about 8, but because of both atmospheric driven ocean acidification, but another process we probably don't have time to cover, but I'll just mention coastal ocean acidification. Okay. The idea that you can get acidification driven by other nearshore processes, that's clearly going on at this site because the levels you can see are getting down as low as 7.5, right? But the kelp is keeping that up. And I'll just mention, you can see there's a lot of wiggle here, and that's actually perfectly natural. What that represents is by day, the pH being driven up by photosynthesis, and then it's sagging down at night. But by and large, during the experiment, higher pH within the kelp compared to off of the kelp. And then in the next slide is the growth rates of juvenile oysters. And so you can see, compared to the ones that were on the kelp or in the kelp, many-fold greater than the control site. And even the ones next to the kelp didn't do as well as in the kelp, but did better than the control site. And we attribute this to, I cite the paper here, again, it's also in the journal Frontiers in Marine Science. You can get online, international peer-reviewed paper. Parallel with this, we also demonstrated in the lab that you can combat ocean acidification with kelp, and that benefits the shellfish. And then I think there's just 2 more slides, I think.
Good than that, yeah.
Yeah. And so the other thing that is not intuitive, but we've discovered through the years, is seaweeds can fight these harmful algal blooms. And so the one that we worry about the most, or I worry about the most in marine systems, is this one right here, Alexandrium. It makes saxitoxin, that's a neurotoxin that can get in shellfish. And there's been, I think, more than 6 or 7 locations across Long Island where they've had to close down the shellfish beds because this toxin gets into the shellfish. And so the question is, can kelp do anything about this? And again, on first blush, one might think, well, that, how could that be possible? But if you go to the next slide, they say a picture's worth 1,000 words, and the name of this picture might be Alexandrium spilling its guts, because essentially what it shows is when exposed to the kelp, the individual cells go through something called chlorosis, so they lose that brown chloroplast that they have, and they just start falling apart. And so in this study here, again, you can look that up and get it online. What we demonstrated is not only do the cells fall apart, but by putting kelp out, say in an experimental setting, we've been able to keep these Alexandrian blooms at bay and even reduce the amount of toxin that ends up in shellfish just by having the kelp there as well. So definitely environmental benefits to go along with what we hope to be the economic benefits as well.
So we're almost out of time, but just to wrap it up.
Sure.
First of all, people have to understand that restoring the bays, mitigating these problems, takes time.
Yeah.
Decades. You've spent decades.
In some cases, yeah.
And that there is no one solution.
Yeah.
All these things are working together to basically change or mitigate the inputs that our population has put in.
Yep.
Time, patience, money.
Yeah. One thing I'd add in there is something term I use is adaptive management, right? And so the idea is, we think we know how things are going to go, but sometimes it doesn't work out, and you have to change course. And that's what science is, right? Science is learning, and based on findings and data, you come up with a new hypothesis, or you modify your hypothesis.
You start with a plan and you stay flexible.
Exactly, and that's so in addition to just yes, it takes a while but it also takes, it take and I guess it would also say it takes good data to go with that, right? So the restoration, but then the monitoring to say yes, it's working, or no, this isn't what you expected, and therefore now time to change course a little bit or do something just a little bit different.
Gotcha.
Yeah.
Well, I'd like to thank you for sharing your work with us. I think this gets to your third goal, sharing your understanding and discoveries with the public.
Yeah.
So hopefully we'll help you achieve that. I enjoyed this conversation. I
Yeah,
hope
it was
that our
fun.
viewers did, or will. I'd like to say that this series is brought to you by the South Fork Sea Farmers. We're a non-for-profit focused on the restoration of our waters. We work collaboratively with the East Hampton Town Shellfish Hatchery to expand and broaden educational programs and encourage residents to be stewards of their marine environment. For more information, visit our website, southforkseafarmers.org. Thank you very much.